from some of that basal part of the brain that sits in the center of the brain, surrounding the third ventricle and just above the hypophyses, pituitary stalk, and is organized into nuclei, paired nuclei, around the third ventricle. Okay. This is a list of the hormones and the regulatory factors that are coming from the hypothalamus. You are going to know these so you can recite them in your sleep, because you will need to know them by the end of the term. ACTH, or adrenocorticotropic hormone, is a product of the corticotrope, which is one of the 5 main cells in the anterior pituitary, and it's involved with control of the adrenal gland, the outside portion of the adrenal gland specifically. 2 of the layers of which are called the fasciculata and radicularis, and are responsible for producing glucocorticoids. Glucocorticoids help balance glucose levels, but as importantly, they're involved in stress responses. ACTH, its controls from the hypothalamus, and glucocorticoids are the stress axis. So when I say stress, I mean everything from heat stress to preparing for exams to being in a concentration camp for a prolonged period of time, to not being able to find water. Any of those things are stressors. To fighting with your neighbor, your boyfriend, and maybe even your girlfriend. Okay, so that's the stress axis. The major factors involved, the ACTH, by the way, is a 39 amino acid long polypeptide that is clipped from a longer precursor that you're going to hear about again, called pro-opiomelanocortin, POMC. Corticotropin-releasing hormone is a small protein. I can never get them straight. I believe that one's the one that's 41 amino acids long. It's produced by cells in the hypothalamus and is dumped into the primary and secondary portal plexus, where it acts at the level of the anterior pituitary on corticotropes. It's stimulatory. Interleukin-1, which is a protein that's produced in the periphery by white blood cells in response to the stress of infection, is also a stimulator of ACTH. Glucocortical steroids are a negative feedback inhibitor of ACTH release and POMC production. And then you've got vasopressin and this polypeptide, PACAP, which stimulate ACTH production. I've got a little article that I'll show you later on what PACAP is, because it's involved in several of these systems. But you've got multiple inputs. They're arranged, by the way, on this diagram in order of importance, okay, in terms of their control. So CRH and interleukin as stimulators are as important or more important than glucocorticoids in controlling ACTH production. Yes?
Is this one of the slides?
Pardon?
Is this one of the slides?
This is actually from, this is one of the slides, but it's actually, what I'm showing you is part of the second PowerPoint presentation series. Okay. Okay, so that's ACTH. The second one on this list is thyroid stimulating hormone, TSH. This is a 2-subunit protein hormone. It's an alpha subunit and a beta subunit that's produced by the thyrotropes, and that stimulates thyroxine production by the thyroid. Now the thyroid act and TSH thyroid hormone releasing, or thyroid releasing hormone, and thyroxine and triiodothyronine constitute the thyroid. Now I know why they're in there. Thyroid axis. Okay. The thyroid axis is involved in maintaining thermogenesis, that is heat production in the organism or organisms. So those organisms that maintain their own body heat will have an active thyroid axis. It's also involved in maintaining growth and development of most tissues. You may recall I mentioned there were 4 hormones that were absolutely essential if you were going to put cells into tissue culture and try to maintain them without serum in that culture. One of them was glucocorticoids, like cortisol. Another one was thyroid hormone. A third one, see if anybody remembers what the third one was insulin, which actually at high concentrations is substituting for a number of other growth factors. And the fourth element happens to be an iron carrier protein, which will make sure the cell can use iron. So you've got the thyroid axis here, and notice here in the thyroid axis you have thyroxine from the thyroid acting directly on the thyrotrope to suppress thyroxine production. You've got a small, that one, thyroxine, is a derivative of tyrosine, so it's an amino acid derivative. TRH, thyroxine releasing hormone, also called thyroleibrin, is a 3 amino acid long peptide with modified N and C termini. And it is stimulatory on the thyrotrope for the production of TSH. Somatostatin. Has anybody ever heard of somatostatin before? If you had read all of the exam questions, you would see somatostatin. Somatostatin is a 14 amino acid long peptide from the hypothalamus. And interestingly, wherever somatostatin seems to work, it's always suppressive. And it suppresses TSH production from the thyrotrope. But it's being produced, in this instance, up in the hypothalamus. It turns out that somatostatin is also produced by some cells in the pancreas, in the pancreatic islets. Luckily, or for whatever reason I should say, the amount of somatostatin produced in the pancreas is insufficient when it's diluted and fully in circulation to act at the level of the thyrotrope. So even though it's produced in one place, there's not enough of it being produced, or it's being degraded too fast, so that it never acts at the level of the hypothalamus or pituitary. Let me skip prolactin for a minute, because it turns out to have some unique characteristics. Somatotropin, or growth hormone, is a protein. It's a single peptide protein made by the somatotrope. And as you found out in the exam, there are two things that help control it. One is growth hormone releasing hormone, or somatolibrin. This is 44 amino acids long. It's a small protein made by cells in the hypothalamus that are coming down through the primary and secondary portal plexus and acting on the somatotropes in the anterior pituitary. It's stimulatory. Somatostatin, our old friend up there, is coming down and suppressing growth hormone production. And you have our friend from up further, PACAP, also stimulating production. So there's a number of things going on. The growth hormone axis, or the growth axis, so you've got GH, and you've got GHRH, and somatostatin controlling production. You'd anticipate something being produced out in the periphery. And what it appears to be is that it's a combination of metabolites that are providing the feedback, the negative feedback, that is primarily responsible for regulating growth hormones, releasing hormone, and production. So these tend to be things like glucose, fatty acids, and certain amino acids. And these overall constitute the growth axis. Somatotropin, GH, is involved in body growth. So during maturation during childhood, you're seeing the length of long bones being stimulated by growth hormone to increase in length, and in support of intermediary metabolism. There's a lot of control of what goes on in the liver, and part of that has to do with production and release of glucose and use of amino acids. So growth hormone axis is intermediary metabolism and growth. The next two are produced by one cell type. So follicle stimulating hormone, or phallotropin, FSH, and luteinizing hormone, or lutropin, are both produced by the gonadotrope. They, like TSH, have an alpha subunit and a beta subunit. We'll talk a little bit more about them in a couple of lectures. The alpha subunit is common among all those three hormones within a given species. The beta subunits are what determine whether you've got LH or FSH or TSH. These two, being produced by the same cell type, are under different controls. Both of them are under a positive or stimulatory control by gonadotropin-releasing hormone, which is also known as luteinizing hormone-releasing hormone, or gonadolebrin. So GnRH and LHRH, same molecule. 10 amino acid long peptide. So this molecule here and this molecule here, same molecule, both stimulatory, stimulates both production of both of those from gonadotropin. And it does it in both the male and the female. Sex steroids, testosterone, estradiol, to a lesser extent progesterone, all are negative feedback inhibitors of production of LH and FSH, usually by controlling production of GnRH. And in matter of fact, there's one thing we've left out here and show up on some other figures. There's a peptide that's produced in the brain and in the upper portion of the hypothalamus called kispeptin, which is a small protein, which is also stimulatory production of this, ultimately stimulatory to these two hormones, but suppressed by sex steroids. There is an exception for sex steroid inhibition of LHRH, and it occurs only in the adult female. And that is when estradiol, or when the female is near the middle of her ovarian cycle, just before ovulation. The production of kispeptin switches from being suppressed in the presence of high levels of estradiol to being stimulated. And as a result, kispeptin goes up, LHRH goes up, and LH goes up, along with FSH. But that's only in the female, only near mid-cycle. So in a prepubertal female or in a post-menstrual female, a female that's after the age of having menstruation or ovarian cycles, she will have a negative feedback even from fairly high levels of estrogen. Okay. Now, there's one other player that's mentioned here, but not here. Anybody spot it? Inhibin. Inhibin. Inhibin is a 2-subunit protein. It's a fairly small protein. The subunits are cross-linked, so they're covalently interlinked. Exactly which one, which kind interacts with another kind of subunit determines what kind of inhibin you're looking at. There's an alpha inhibin and a beta inhibin. The alpha inhibin is made by females during the luteal phase of during the latter half of their cycle. Beta inhibin is made by males, and it's made by females in the early portion of their cycle. Inhibin suppresses directly the gonadotrope, so no involvement of the hypothalamus. So this is a direct inhibitor of FSH production and release at the level of the gonadotrope. Notice that isn't occurring down here with the gonad or the LH. That's got the other common controls, but the inhibit unique. Okay. FSH, follicle stimulating hormone, is going to support the early growth of ovarian follicles and estradiol production by those follicles. And in the male, it supports Sertoli cell function and spermatogenesis. Sertoli cells are what will be called nurse cells. They're intimately involved in production of all stages of sperm, and they respond to FSH. There are a couple of other responses we'll get into and when we talk about that in more detail. Luteinizing hormone supports late follicle development, ovulation, and this temporary organ that forms from the follicle after ovulation is called the corpus luteum. The corpus luteum is normally a storehouse and production site for progesterone. It's the main site for progesterone in mammals. In the male, LH supports testosterone synthesis in a cell called a Leydig cell, which lies outside the structures that are involved in sperm production. In the testis, but outside the cell, outside the sperm production area. So these are all involved in reproduction. Okay. The last one on the list, we'll get back to prolactin in a minute. The last one on the list is melanocyte stimulating hormone, MSH. There are actually about 4 flavors of MSH. So there's alpha, beta, and gamma. I think there's one more. They're all about 14 somewhere between 10 and 14 amino acids long. They're later products of metabolism of pro-opiomelanocortin, which you ran into or heard about at the start, ACTH production. So these are pieces, other pieces of the same starting material that you get ACTH from. The production of melanotropin is stimulated by our old friend corticotropin releasing hormone, CRH. And MSH goes out and acts peripherally on melanocytes, primarily. Careful on that, melanocytes. Melanocytes are pigment-containing cells in the integument. So they're in the skin, they help, in the long run, they will determine pigmentation of the skin, of the hair, of feathers if you have them, the coloration of your scales if you happen to be a lizard or a fish. So all of that is supported by MSH. The other thing that they're involved in, which may be more important in mammals at any rate, is alteration of the adrenal response to ACTH. They turn out to also be involved in control of appetite and glucose metabolism, the actual control of body mass. So, not unimportant, but where are the melanotropes? Does anybody remember? They're in the intermediate lobe. Okay. The intermediate lobe of the pituitary lies where? It's the interface between the posterior pituitary lobe and the anterior pituitary lobe. It persists in fish, in reptiles, in birds, and in some mammals, but not primates. So adult primates, including humans, don't have anything more than maybe a thin layer of cells that are melanotrophs. Most of the MSH you see in primates is coming from later metabolism in corticotropes and in the central nervous system, because it's also produced there. All right. So, hormones affecting lots and lots of different systems. Prolactin. Notice that most of the primary controls are stimulatory, and feedback controls from the periphery are negative. Under those circumstances, bad picture. Under those circumstances, you're getting a stimulus from the hypothalamus to the production of a protein hormone that goes out into the periphery and stimulates something else, or the target. And the target's responses generate a negative feedback. And that negative feedback often occurs at the level of the hypothalamus, but can occur also at the level of the pituitary. So this is hormone 2, or metabolite. Now, under these kinds of circumstances, what would happen if I broke the stalk of the pituitary? What would happen to most of these hormones?
They're not going to be produced because there is no signal coming from the
Okay.
hypothalamus.
If you're lacking this positive signal, you're probably not going to produce the hormones. So what does that include? ACTH, TSH, growth hormone, follicle stimulating hormone, LH, and melanotropic. If I break this stalk and don't do anything, what am I going to do to that organism or animal? So if this is a clean break, or there's something that's driven in here to cut that circulation off, is that animal going to live?
No.
Probably not. It may persist for a while, but what's it lost?
Stress response.
You've lost control of glucose metabolism, you've lost thermogenesis, you've lost supports on intermediary metabolism, anything to do with reproduction, and potentially controls on feeding behavior. Yeah?
These are the two lobes of the pituitary.
These are the posterior pituitary, and this is the anterior pituitary. And if you have intermediate lobe, it would be here.
Second on top.
Pardon?
What is the?
What's this thing up here? Hypothalamus. That's the source of those signals. So, maintain an animal without a pituitary if I'm very careful to keep it warm, provide it adequate water, provide it glucose at appropriate levels, and to make sure that its diet contains appropriate amino acids, fats, sugars, anything it needs. And so long as I keep that animal calm, it's a calm environment. I don't want to get it excited. I don't want to stress it in any way. Under those circumstances, I can maybe maintain them a month, a couple months. Right. But pituitary function is central to homeostatic controls in lots of different systems. Yeah?
This is similar to like when you're saying like if you do a clean cut there,
Yeah.
you'd have a break in that negative feedback, right?
You'd have a break in the negative feedback with the possibility, well, yeah, you'd even have a negative feedback broken for TSA, or for T4, because that's still coming in through the stalk.
Right, and same with ACTH.
All those controls from the hypothalamus are coming in through that limited blood supply up here. Now, there is one exception to all these hormones from the anterior pituitary going down, and that is prolactin, which is also known as mammotropin or luteotropin. It's coming from the lactotrope or the mammotrope, same cell type. And that's because the primary control on production of prolactin is negative. It's dopamine. So dopamine coming down this track is having a negative effect on prolactin production. So in order for prolactin production to go up, you normally have to inhibit dopamine production in the hypothalamus. Now dopamine is a neurotransmitter, it's an amino acid derivative. It's produced in at least 10 different circuits in the brain and the body. This is one of those tracks. So you may have to suppress dopamine in only that one track in order to get control of prolactin or get prolactin to go up. Prolactin will also go up in response secondarily to thyroid releasing hormone. It tends to be suppressed by somatostatin, and it's stimulated by estrogens and by oxytocin.
So in a woman who just
Gave birth,
all those hormone levels are
high. A woman who just gave birth definitely is going to have high oxytocin levels. She will have been exposed to estrogen levels that are high for a prolonged period of time. Part of the reason estrogens are stimulatory is because they cause multiplication of lactotropes. And you'll find out that estrogen in many instances is mitogenic. It stimulates cell division and multiplication. So part of this positive effect has to do with actual increases in responsive cell numbers. Yeah.
But would that also mean that like there's low dopamine and also high, or are they all depending on?
They're all, okay, okay, at the end of pregnancy, high. This was high, it's going to go down because the placenta was the normal source of that estrogen. But the effect on cell growth is already there. Okay. Somatostatin, not so much of an effect. TRH, this axis turns out to be stimulated during pregnancy as well. So that's probably high. But the important thing is that you're suppressing dopamine just after birth. And you're getting, and dopamine is being suppressed by a neural circuit, okay? Once the offspring starts suckling, there's a tactile response to stimulation of the breast. And that tactile response stimulates oxytocin, or pardon me, suppresses dopamine centrally. And as a result, you get an elevation of prolactin.
So does that cause any like neurological differences? Like, because dopamine levels would be decreasing? Like have they?
Remember, it's one of 10 different circuits, okay?
So it's not significant.
It's not necessarily significant because you're getting to that one circuit. So prolactin is the one exception. So even if I were to take the anterior pituitary particularly and move it elsewhere, move it under the kidney capsule where I can keep the tissue alive, that animal is going to have an elevated prolactin because it's lost its controls. But the other hormones will have fallen, and you'll see the effects of those falls. I've still got a few minutes going. So where do these come from? There's sources of 3 of them. These are pictures taken of brain, of the forebrain. It's cut through, it's called the sagittal section. It's cut across the brain. This is the optic chiasm. This is the crossing point for the optic nerves. Here's the 3rd ventricle. And these are various nuclei or concentration points for nerves that produce various peptides in the hypothalamus. What's being shown here for thyroid releasing hormone is that it starts in the paraventricular nucleus and the fibers kind of loop up and swing down past the optic chiasm toward axonal ends on or in or near the primary portal plexus, near the upper end of the stalk of the pituitary. The suprachiasmatic nucleus is also going to contribute to these fibers coming down here. This is the part of the magnus, pardon me, these are part of the tract that's going to control particularly thyroid hormone, but recognize that TRH also contributes to control of prolactin, and what else? I think it was growth hormone. Growth hormone releasing hormone, you're seeing it come from other nuclei. Notice this is the optic chiasm. This is further back behind this in the brain. The median eminence is essentially the stalk of the pituitary. Your dorsal median hypothalamic nucleus, the ventromedial hypothalamic nucleus, and the arcuate nucleus particularly seem to be sources of that particular protein hormone, and they're all meeting at the level of the median eminence. Recognize that these are shown for 1/2 of the brain. There would be a similar picture on the other half because these are paired nuclei. There's one over here and there's one here, one here and there's another one here. Did you get the picture? Okay. So those are all coming down in this area. And then finally, LHRH. This is shown for primates particularly, where most of the LHRH producing neurons are just above the median eminence in the area just below the arcuate nucleus. If you were looking at primates, there is another set of LHRH producing neurons that are up further forward in the supraoptic nucleus. Okay. The reason we know that is because a lot of these early experiments that teased out where these were coming from were careful experiments that came in and cut nerve tracks. And one of the ones that was fairly accessible was these preoptic area LHRH nerves. So if you cut just below there, you cut the source for the LH surge in females, but you still saw LH production and LHRH production in both males and females because there was this other population of the hormone producing neurons. Okay, so they come from a variety of nuclei, paired nuclei in the hypothalamus around the third ventricle, and they're feeding down through the primary and secondary portal plexus onto the cells in the anterior pituitary. Okay. Now, we're going to take you through circuits. Okay. Get used to circuits. This is simply showing you the hypothalamic nuclei as a block. I wasn't particular in where I put a particular hypothalamic nucleus. And here's the thyroid choke down in the anterior pituitary. This cell is producing thyroid stimulating hormone, which is going to act at the level of the thyroid tissue, specifically the thyroid follicle cells that produce thyroxine and triiodothyronine. Okay. The control, as I told you earlier, stimulated by thyroid releasing hormone and suppressed by somatostatin. The primary control for this whole loop is actually right here. This is actually sometimes called the short loop feedback because it only involves two cell types, the thyrotrope and the thyroid follicle cells. That's the major control. So if you wanted to make this more dense, that would be the major control. These are secondary. So that's the control of TSH. And ultimately, T3 and T4 are going to affect intermediary metabolism, thermogenesis or heat controls, and growth. That one's pretty easy to learn. So what are the impacts? This shows you an individual who lives in a portion of the New Guinea highlands that has endemic iodine deficiencies. So the diet lacks iodine, and you need iodine to make thyroid hormones in the pituitary, or in the thyroid gland. If you lack it, you've broken the feedback loop. Let's see if I can go back. If you lack iodine, you've broken this feedback loop because you can't make thyroxine. So this is broken. This is broken. So what's happening? This is stimulating the tissue to try to make that product. But if you can't make the product and there's no feedback, the signal is keep making it. Keep stimulating it. Now, trophic hormones. A lot of these hormones are called tropic or trophic hormones. What does tropic refer to? No.
They affect a different sound.
No. Trophic or tropic refers to growth, cell growth. They stimulate cell growth. They will stimulate their targets to do a variety of things, including to grow. So TSH stimulates not only production of thyroxine, but it will stimulate growth of the tissue. It's trying to make the tissue. When we get into talking about the thyroid axis, you'll find out that there are storage deposits of what's going to be thyroid hormone. And those storage deposits get greatly enlarged, but since you haven't got any iodine, you can't actually make the hormone. The end result of that stimulation is this formation of what's called a non-functional goiter. It's non-functional because it can't make thyroid hormone. Yeah?
So that swelling there, that's the result of extra tissue, that's an extra thyroid gland?
Correct.
Okay.
Okay. It's a growth of the thyroid gland. It's the abnormal growth of the thyroid gland. It's not making, but it's not making thyroid hormone here. We can get to a condition like this when thyroid is being produced, when you have a disease that stimulates thyroid stimulating hormone receptor without the thyroid stimulating hormone. Okay. And in that particular condition, you get production of thyroid hormone. It shuts down TSH and TRH production. But the stimulator, which turns out to be an antibody, an autoantibody, is not under the control of thyroid hormone. So it continues to stimulate the gland. So the gland gets big like this. It produces thyroid hormone, and you see all the effects of overproduction of thyroid hormone, but you don't see any normal production. And you have to do immune suppression to bring that autoimmune antibody down in order to control the situation, rather than doing anything to deal with the thyroid axis itself. Okay. ACTH, the stress axis. Here we're dealing with the corticotropic producing adrenocorticotropic hormone, and it's acting on the outer layers of the adrenal gland, the steroid producing ones, to produce glucocortical steroids, which affect metabolism and growth. Glucocortical steroids have a negative feedback here and help suppress ACTH via control on corticotropin releasing hormone production. They're also suppressing immune function and the production of IL-1 beta, which normally would be stimulatory up there. And then you've got vasopressin, vasopressin, which is, remember, an posterior pituitary peptide hormone, which contributes to stimulation of ACTH. And then various stressors acting through the nervous system to stimulate, particularly CRH production, to stimulate ACTH production and glucocorticoid levels. So when you have many disease states, particularly if you get into an autoimmune type condition, they will often they will look at your glucocorticoid levels in the lab, but they will also potentially give you glucocorticoids to suppress your immune response, to get things back to normal. So here we have a connection between the glucocorticoid axis and the immune system. One of the things you're going to see all along is you're going to start to see that all these hormone systems, all these hormonal control loops, feed into or interacting with other physiological systems. When we look at a physiological system in immunology, in endocrinology, in neurobiology, we use what's called a reductionist approach. We look at a simplified picture. In matter of fact, evolution acts on the whole organism. So it's how is this thing integrated? So it's not just the immune system acting alone, it's not just the endocrine system acting alone, it's all of them acting together. So you'll see connections all the way through this set of information. We'll finish this up next time and move on to our next topic. Okay, anybody here who took the exam on Friday needs to pick up an exam? It's up here. Thank you.